I was a clinical fellow at Memorial Sloan Kettering. Had done a bunch of research in the past and wanted to continue doing research that had to do with cancer. And I was looking for labs, shopping for labs and for mentors. And I was fortunate that Craig Thompson took me in. And so when you start as a postdoc, you're new and you're shopping around for ideas and you're shopping around for fellowships. And there was another postdoc in the lab, Wilhelm Palm, who was also a Hope Funds fellow. And so I sort of heard about the Hope Funds fellowship from him, and he encouraged me to apply. And I applied, and I was lucky to get it. The traditional textbook view of metabolism in cells is metabolism has to do with what cells eat and how what they eat allows them to make the things they need to make, right? So if they need to make more membrane or more nucleus, or they need to divide, they need to eat certain sugars or eat certain lipids, etc. And that has been sort of the classic idea of metabolism. And one of the things that came out, some from Craig's lab, some from other labs, is that cells will also use what they eat to carry out decisions. And what I mean by decisions is, for example, there's a phenomenon known as differentiation. So cells can become, so stem cells, for example, cells that are, let's say, very early on in their lifespan, and they are able to become many different things. So the classic example is an embryonic stem cell, like the first cell of the embryo is one cell, and that cell has the ability to become all the future cells of the body. And that process is known as differentiation. And classically, we always thought that that what you become, so whether a cell becomes a muscle cell or whether it becomes a fat cell, has to do with what genes you turn on. And you turn on genes by using certain proteins called transcription factors to turn on certain subsets of genes versus other subsets of genes. It turns out from what work that had been done in the lab before that what a cell eats can also dictate what it becomes. So you are what you eat in a way. And that was a very interesting idea for me for two reasons. One, because it's very interesting, but also one of the phenomena that happens in cancer is that, so in our body there are these stem cells in most of our tissues whose normal role is to differentiate. So there's stem cells in the bone marrow, for example, which normally will divide and then commit to a certain fate. So either they will become red blood cells, or they'll become neutrophils, or they'll become platelets. And one of the things that happens in cancer is that phenomenon, the differentiation, is blocked. And so one of the interesting ideas about studying how metabolism affects differentiation is that metabolism and metabolic enzymes are very immediately druggable. And so one could come up with ways to, for example, if you have cells that aren't able to differentiate, to relieve that block and allow them to differentiate. And so there was a very immediate therapeutic opportunity, I felt. And it was an embryonic stage in a field where I sort of wanted to go. I work on a set of mutations that happen in a variety of different cancers. They were originally described in leukemia, but they're present in some sarcomas. They're also present in a subtype of liver cancers. And they're mutations in a metabolic enzyme that traditionally was thought to be involved in how cells use sugars to make energy. But it turns out these mutations produce a molecule that leads to a block in differentiation. So normally these cells would be present in the bone marrow and differentiate into, let's say, neutrophils. But if they have this mutation, they're blocked and they can't differentiate and they proliferate and cause leukemia. And there are drugs that can reverse that differentiation. that differentiation block. And so I've been looking at how that happens. How does that mutation actually block the process of differentiation? There's been a lot of, in the last 20 years, 15 years, there's definitely been a lot of different doors have been opened in terms of how we think about reversing the changes that happen in cancer cells. For sure, there are a lot of things we haven't looked at and we haven't thought. I think we tend to jump on bandwagons of what's cool and interesting at the time. And so there are always fields that are left aside. And then you look back at the literature 10 or 20 years ago, and you realize that people were working on some things that become suddenly interesting because of what we know now. And I think that's a constant cycle. I think the one sure thing about science is that you have to hit yourself against the wall many, many times until you come up with something interesting. You have to be excited about what you do. And I think it's a mix between offering ideas that are interesting, that are new, and that are doable. And there's a balance. It's sort of like you can't do all of those three perfectly. And there's a balance depending on what organization you're asking for funding on what you have to stress. So there are certain places where they really want to fund things that are doable, and there are other places that want to fund things that are more risky, but where it's sort of a high risk, high reward. The Hope Funds has made an effort to fund, first of all, people in really good labs, which already, in a way, selects for good candidates. They have a very good track record of where the former fellows have gone, but they also, I think, fund risky ideas. So ideas that, in my experience, were seen as too risky in other organizations, where here they were welcomed. And I think that's personally rewarding, because sometimes you have, especially when you start, you're more ambitious and you say, well, I want to try this really cool thing, and then someone says, oh, that's never going to work, and you want people to say, well, let's try it. I wear two hats, and it's very hard to wear both of those hats at the same time. So I'm a clinician. For a year now, I've been an instructor, which essentially means I have my own clinic. I see patients with gastrointestinal malignancies in the GI oncology service at Memorial Sloan Kettering. So I see patients with colorectal cancer, I see patients with pancreatic cancer, and with cholangiocarcinoma, some with anal cancer also. And so I see a mix of patients that have very aggressive, uncurable disease that we can manage, treat, but where there certainly is a need for better opportunities. And then I see other patients that we cure with current treatments. And so that is very challenging, but it's also very rewarding in an immediate way. And I do that half a day a week. And the rest of the time I'm in the lab. And in the lab, I do a mix of asking some very clinically related questions. But most of the questions that I ask are very basic and have even more to do with how cells make decisions about what they want to be. And that's very important for cancer, but I study the basic question in and of itself. And I think I think that mix is key, sort of studying basic biology. You will learn things about cancer that you wouldn't have thought of if you only look at a cancer cell. Because ultimately what a cancer cell does is use the toolkit that our cells have, our normal cells have, to invade, proliferate, metastasize. So going back to your question, my ambition is to ultimately have my own lab where I can continue wearing, I said two, but it's probably three. It's sort of the clinical hat, the translational hat, and the basic science hat, and trying to fuse those three aspects in a way that makes sense, and to continue enjoying doing that. Ultimately doing science is just it's basically like constantly solving a puzzle. And so if you like solving that puzzle, you will bang your head against the wall, but you'll continue doing it. And so it's a pleasure to be. It's a responsibility, but it's also very rewarding when it works.
When it comes together.
Right.